Welcome to today's video where we will be looking at the cell cycle, cell junctions, and cellular adaptations. Remember to like, share, and subscribe to my YouTube channel for more amazing videos. One cell cycle consists of two phases, the interphase and mitosis. Interphase is the phase of the cycle when the cell is not dividing, while mitosis is the division of genetic material. Cytokinesis divides the cytoplasm, producing two distinctive cells. Interphase has three phases. A cell grows and carries out all normal metabolic functions and processes in a period called the first gap phase. The first gap phase is followed by the S phase, where there is replication of DNA. After the S phase, the cell goes to the second gap phase, where the cell continues to grow and make necessary preparations for mitosis. The first gap phase varies in duration, from hours to days. The S phase lasts 8 to 10 hours, and the second gap phase lasts approximately 5 hours. Some cells don't proceed to mitosis after the interphase, but enter the resting phase. Cells that have temporarily stopped dividing, cells in resting, and cells that have permanently ceased dividing, for example, nerve cells, are said to be in the resting phase. The mitotic phase takes 1 to 2 hours. During this phase, a cell undergoes two processes. The contents of the nucleus are distributed between its two halves. Cytokinesis then divides the cytoplasm and cell body into two cells. Mitosis is divided into four phases. The first is prophase, where the chromosomes condense and become visible. The spindle fibers emerge and the nuclear envelope breaks down. The centrosomes also move to the poles of the cell. The second stage of mitosis is the metaphase stage, where chromosomes line up at the metaphase plate. Each chromatid is attached to a spindle fiber originating from the opposing pole. The third stage of mitosis is the anaphase, where the centromeres split into two and the sister chromatids are pulled towards the opposite poles. The spindle fibers also start to elongate the cell. The last stage is telophase, where chromosomes arrive at opposite poles and begin to condense. The mitotic spindles break down and the nuclear envelope surrounds each set of chromosomes. Cytokinesis then breaks the cytoplasm and cell membrane into two cells. Unlike mitosis, which is complete after a single round, meiosis occurs in two successive stages. Meiosis 1 consists of four phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis begins with a diploid starting cell and ends with two cells, each with the haploid number of chromosomes. The second stage of meiosis, meiosis 2, also consists of four phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. During meiosis 2, each of the two haploid cells formed during meiosis divides, and the net result is four haploid gametes that are genetically different from the original diploid starting cell. Cell junction is the connection between the neighboring cells or between the cell and extracellular matrix. It is also called membrane junction. Cell junctions are classified into three types: occluding junctions, communicating junctions, and anchoring junctions. Occluding junctions are cell junctions which prevent intercellular exchange of substances. They are also called occluding junctions. These junctions prevent the movement of ions and molecules from one cell to another cell. Tight junction is the intercellular occluding junction that prevents the passage of large molecules. It is the region where the cell membranes of the adjacent cells fuse together firmly. Communicating junctions are cell junctions which permit the intercellular exchange of substances. These junctions permit the movement of ions and molecules from one cell to another cell. Gap junction and chemical synapse are the communicating junctions. Gap junction is the intercellular junction that allows passage of ions and smaller molecules between the cells. Anchoring junctions are the junctions which provide strength to the cells by acting like mechanical attachments. These junctions provide firm structural attachment between two cells or between a cell and the extracellular matrix. Anchoring junctions are responsible for the structural integrity of the tissues and are present in the tissues like heart muscle and epidermis of skin, which are subjected to severe mechanical stress. The firm attachment between two cells or between a cell and the extracellular matrix is provided by either actin filaments or the intermediate filaments. Depending upon this, anchoring junctions are classified into four types. Where actin filament attachment provides a cell-to-cell attachment, the junction is called an adherence junction. Where actin filament attachment provides a cell-to-matrix attachment, the junction is called a focal adhesion. Where an intermediate filament attachment provides a cell-to-cell attachment, the junction is called a desmosome. Where an intermediate filament attachment provides a cell-to-matrix attachment, the junction is called a hemidesmosome. Cell adaptation refers to the changes taking place in a cell in response to environmental changes. Normal functioning of the cell is always threatened by various factors such as stress, chemical agents, diseases, and environmental hazards. Only during extreme conditions does the cell fail to withstand the hazardous factors, and this results in destruction and death of the cell. Atrophy means decrease in size of a cell. Atrophy of more number of cells results in decreased size or wasting of the concerned tissue, organ, or part of the body, and can be caused by poor nourishment, decreased blood supply, lack of workload or exercise, and loss of control by nerves or hormones. Hypertrophy is the increase in the size of a cell. Hypertrophy of many cells results in the enlargement or overgrowth of an organ or a part of the body. Hypertrophy is of three types: physiological hypertrophy due to increased workload, pathological hypertrophy in response to pathological changes, and compensatory hypertrophy, which compensates for the loss or dysfunction of another organ of the same type. Hyperplasia is the increase in the number of cells due to increased cell division, resulting in the gross enlargement of the organ. Physiological hyperplasia is the momentary adaptive response to routine physiological changes in the body, for example, the endometrium during menstruation. Compensatory hyperplasia is the increase in the number of cells in order to replace the damaged cells of an organ or the cells removed from the organ. Pathological hyperplasia is the increase in number of cells due to abnormal increase in hormone secretion and is also called hormonal hyperplasia, for example in gigantism. Dysplasia is the condition characterized by the abnormal change in size, shape, and organization of the cell. Dysplasia is not considered as true adaptation, and it is suggested as related to hyperplasia. It is common in epithelial cells of the cervix and respiratory tract. Metaplasia is the condition that involves the replacement of one type of cell with another type of cell. Pathological metaplasia is the irreversible replacement of cells due to constant exposure to harmful stimuli. If a cell fails to adapt, it dies. Apoptosis is defined as the natural or programmed death of the cell under genetic control. It is also called cell suicide, since the genes of the cell play a major role in the death. Necrosis is the uncontrolled and unprogrammed death of cells due to unexpected and accidental damage. It is also called cell murder because the cell is killed by extracellular or external events. Thank you for watching this video. Remember to like, share, and subscribe to my YouTube channel for more amazing videos of anatomy and physiology.